This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 212, recorded on January 30th, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm well. How are you?
Okay.
How's San Diego?
Fantastic, as always.
It's very cold here. It's freezing.
Oh, boy. Well, actually, we complained that it's cold here because it's 63 degrees.
You know, here it's 1 degree Celsius, just above freezing.
God.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. It is -1 Celsius here today.
Wow.
Oh, God.
You guys have snow?
Really, it's been melting. We've been above freezing every day for about a week, and it's going to be in the 40s early next week. So I can't complain. I can worry, but I can't complain.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. It's 13 degrees C, 55 Fahrenheit here in beautiful Charleston.
That's nice weather.
Yeah, we're going to get a little rain this afternoon, I think.
Wow. That's the place to be, Charleston, except when there's a hurricane.
Except when there's a hurricane or an earthquake. We had a little tremor. We felt a little bit of that Florida tremor that everyone's been talking about.
All right, today we have a snippet in a paper. My snippet is going to be a paper recently released about the new coronavirus circulating in China. And I'm sure all listeners have heard about this. It's a topic we would usually reserve for TWiM. And in fact, we have done 2 episodes already. So if you want more detail, you can go there. In fact, particularly last week's TWiM with an expert, a coronavirus expert, Ralph Barrick, was great.
And I heard that report, and it was outstanding. I sent it on to the editorial staff of our local paper who's writing an editorial on it, and they found the information to be compelling and factual and non-sensational, and the discussion really helped them with their editorial position. So you did a public service for the greater citizens of Charleston.
That's wonderful. That's what we try and do on these podcasts. We try and provide accurate information and talked about in an intelligent manner. And so,
and there sure is a need for that with this coronavirus. So illuminate.
So I wanted to mention a few things from one of the first papers that was published. It came out last Friday in Lancet. It's called Clinical Features of Patients Infected with the 2019 Novel Coronavirus in Wuhan, China. And this is a group out of various institutions in China, including a hospital in Wuhan and Chinese Academy of Sciences in Beijing, etc. So the publications are now starting to come at a rapid pace. It's hard to keep. I liked the first week when there were just 1 or 2. This one has some very interesting tidbits in it, and we can use this to frame this discussion. As you know, back in December, there were a cluster of cases of pneumonia in Wuhan of unknown cause. A cluster meaning more than just one, because you get pneumonia all the time in the winter, and you usually find out what's causing it, usually bacterial pneumonia. But in this cluster, 59 suspected cases, people with fever and dry cough, they were hospitalized, and they did a battery of clinical tests to find out what they had, and nothing known showed up. None of the known viruses or bacteria turned out to be causing their pneumonia. And so in separate work, they did bronchoalveolar lavage on these patients. And from that, they sequenced it and they found a novel coronavirus. It's also been cultured and grown in cell culture. And the coronavirus, the genome is 29,000 bases. The highest homology is to a bat coronavirus, which was sampled years ago in a cave about 1,000 miles away from Wuhan. So because of the SARS outbreak years ago, which was caused by a virus that came originally from a bat, bats have lots of viruses that circulate in them. There's been a lot of surveillance of bat viruses in China. And so there's a database of sequences. And so immediately they found that this new virus, 96% identity with a bat virus. So that's the closest. It's close, of course, to other bat viruses, SARS-like bat viruses. It's also close to the SARS virus, but it's not those viruses. It's a new virus. Now, this paper is interesting because there are a few tidbits of information that are actually surprising, because originally we were told that the outbreak started on something like December 19th, I believe, and it was reported to WHO on the 31st of December. And the initial cases, they said there was a shared exposure to the Huanan seafood market. So these patients all went to this market, right? And so the market was closed down on January 1st. This market, by the way, sells not only fish, but all sorts of meat products.
It's called a wet market because it effectively has livestock that they'll quickly dispatch for the consumer, and you take home your fresh meats literally after about 15 minutes. It's live and on the hoof, and you go away and they dispatch it, and you come back and you get your fresh meat.
Yeah, and they have all kinds of exotic meats, including bats and civets and raccoon dogs and frogs and all sorts of things. Okay,
and that's relevant. What Michael said is because there would be exposure not only to respiratory droplets but also blood products.
Absolutely, absolutely. And we know that this kind of situation started the SARS outbreak in 2003. It has started outbreaks of avian influenza. So it's a tradition in China, but it's dangerous, especially with the population getting more and more dense and interconnected. Now, in this paper, they studied this group of 41 admitted patients. They all had laboratory-confirmed infection because by now we have the genome sequence. You can make PCR primers, and they were able to see who was infected. And they have a really interesting epidemiological graph where they plot the number of cases with the date starting on December 1st through January 1st. And these are the 41 cases. The first case is December 1st, and that is ahead of the time when this outbreak was originally thought to begin. In that case, one individual infected on December, or showed symptoms on December 1st, had no connection with the Huanan seafood market.
So that's likely patient 0.
That's probably patient 0. Of course, there may have been others we don't know about, but for our purposes, and all the other patients, all the other 40 patients in this study had no connection with that patient, none whatsoever, no contact. And about half of them had contact with the fish market, and the others did not. Maybe 30% did not, 66% did. So there are quite a few patients, 13 I think in all, who had no contact with this Huanan fish market. And so now we don't know where this started. There's a quote in Science magazine this week from a Chinese scientist who's working on this. He said, we really don't know where this came from. It's obviously not the Huanan fish market. It's somewhere else, and we're trying to figure it out.
And the only way we'll know is if we go back and do retrospective serology and look at who's been seroconverted and whether or not they've actually displayed any upper respiratory symptoms.
That's right. That's right. That's exactly what we have to do.
Did that patient, the December 1st patient, have a travel history?
Let's see if they say anything about. I don't remember.
I didn't come across it in
anything about that. No, they don't give any information about that. No, these patients all, by the way, had pneumonia, and I think they all recovered in this particular cohort. So that's very interesting. Common symptoms. The most common symptoms: fever, cough, and fatigue or myalgia. And so those are the main symptoms of this. And that's why you can do thermal scans at airports and pick up people who are into the disease. So the other point here that's very interesting is that if this person got sick on December 1st, he was most likely infected sometime in November.
Mm-hmm.
could have been anywhere from a few days to 2 weeks earlier. And elsewhere, people have been analyzing sequences of multiple isolates of this virus from different people, and you can back calculate to when you think was the most recent common ancestor, and that can go all the way back into October. So it's possible.
And what you may want to mention, Vincent, is that this is a single-stranded RNA virus that's in one as a message. It's very similar in terms of its structure, in terms of being a single piece of positive structured RNA.
Yep. Plus stranded RNA virus, that's right.
Yep.
With an envelope, so it's a bit labile. It's not terribly sturdy. And so the reason I mention this is because the number of cases just a few weeks ago jumped big time, and it wasn't consistent with this early number. There were 40 cases for a couple of weeks, and all of a sudden we had hundreds and thousands. And so people think this virus has been circulating in people probably at least since October or November. And it's maybe caused a case of pneumonia here or there, but as I said, nothing unusual because pneumonia happens in winter. But then all of a sudden, boom, it got to a certain level. And that's why we are now at something like over 7,000 cases and 175 deaths.
So is it likely that once it did get into this market, then we saw an explosion, like a high density of cases traced back to one place, and therefore it became recognizable to epidemiologists?
Probably, yeah. But this one case from December 1 shows that it didn't start there. But yeah, the market still had some role because a lot of people had contact with the market. So, it may have, someone said this week, it may have come into the market and spread further there.
Right.
Because a lot of people go to market. So that's a common place where you could get infected.
And the city is a major transportation hub for that province. It has approximately, what was it said on your podcast last week, approximately 400,000 people go through its train station each day.
Wow.
Yes, big hub. And of course, the Chinese New Year just began. A lot of celebrations and people traveling around. So at the moment, every province in China now has multiple cases of infection with this virus. The other thing I wanted to point out from this paper is that they appear to be doing some antiviral testing. There is a drug called remdesivir. It's a broad-spectrum antiviral, and it's been previously shown that it's effective in treating MERS coronavirus infected people. And so they say here a randomized controlled trial has been initiated quickly to assess the efficacy of that drug and others for treating that. So that would be interesting if that turned out to be effective. Now,
but like they said last week, it's going to have to be administered very similar to the anti-influenza drugs. You're going to have to take the drug early in the course of your infection to limit the spread of the virus within your lung.
That's right. Now, a couple of other things that are important here to consider. So there are so far 7,000 plus cases, 175 deaths. I just want to point out that so far in the U.S., there have been 15 million cases of influenza and 8,000 deaths. So,
But today in Chicago, the first victim in Chicago actually transmitted it from wife to husband.
Yes, that was bound to happen.
Maybe.
Right.
Let me tell you what the New York Times headline says right now. Breaking news. The U.S. now has a case of person-to-person coronavirus transmission.
Right.
Mm-hmm.
That's the Chicago piece of data.
So, I mean, this... I find this a bit amusing because there's way more influenza and it's way more serious and it doesn't make headlines. And though people don't get vaccines, which we have available. And so now people are clamoring for a vaccine for this new virus. And what if we have one and it becomes another influenza? People won't get the vaccine. It's crazy. It's just crazy.
maybe this will bring in some of those people that have been hesitant to be vaccinated and will be more diligent. But I'm also curious of how I've also been reading great reporting in the New York Times that one of the challenges in China is that their healthcare system is under a strain in a good good week. So now with this outbreak, it's very difficult for people to get in and get the care they need. So my question, Vincent, is how translatable are the case fatality data that we get out of China, and are they applicable to a country that's got a more stable healthcare system like our own?
Yeah. So far, the fatality has not been high. It's actually been lower than SARS.
Right.
And I think most of the deaths are in people over 65 with other health care issues, other lung and heart problems, for example. So I do think they're translatable to other countries. I think it's a good sign because most of the infections in young people are very mild. It's just a very mild influenza. And so that's good in a way, but it's also difficult because most people are not going to hospitals. So for SARS, which was far more serious, most of the transmission occurred in hospitals because you got very sick, you were admitted, and then you transmitted it to other people. It was so serious that you didn't walk around shedding virus. In fact, for SARS, the peak shedding is at the peak of illness, and that mostly occurred in hospitals. With this virus, it's far less serious, so not as many people are being hospitalized. Consequently, people are walking around and spreading in the community.
Yeah, this is more like the common cold cousin.
Exactly.
Of this variety
and that's a problem for control. SARS was controllable because we could control infections in hospital, and in the end, the virus was not very transmissible.
Do we as yet know whether or not individuals are infectious before they display symptoms, that is, in their prodromal phase, or are they only infectious when they have symptoms?
I don't think we have the data on that yet.
Now, and that's what Tony Fauci was saying on NPR a few days ago, is that we still haven't seen the data as to whether or not people are actively shedding virus during their prodromal or pre-symptomatic phase.
I suspect they are because there have been enough travel-associated cases where so the first case in the U.S. in Washington, that individual said he didn't see any sick people around him. So it must have been a person infected who was not showing symptoms and transmitted. I suspect that's the case, but we don't have hard data yet.
Unless it survives on inert surfaces.
Yeah, it could.
Fomites. Do we have a feel for how many people have been exposed but handled the infection without symptoms?
Well, it's far more than have been diagnosed, right?
Right, yeah. Right.
At least 10 times more, typically.
Right.
So, in fact, China's tracking a lot of people who have had contact with people with symptoms, but there are far more people who are infected, and that's part of the problem. People are...
and it might be part of the good news that a lot of people can handle it?
Yes, I think that is good news.
Right.
I said to a reporter, the good news is that if you're under 65... it's mild. And she said, well, it's not good news if you're over 65, is it? I said, no, but there's always a silver lining to everything, right?
Right.
Yeah. So. And in the week since that Lancet paper came out, another manuscript came out of the New England Journal of Medicine this week, reporting on the first 425 patients, where again, it confirms the median age of individuals getting the disease were 59 years old, and 56% of them were male. And then they go on to report that the mean incubation period, or the time it takes you to display symptoms, was 5.2 days, with a range between 4.1 and 7, with the 95th percentile of distribution at 12.5. And the reason everybody is so concerned is that in the early stages of this outbreak, they're referring to it as the epidemic doubled in size every 7.4 days, which suggests that the basic reproductive number, or how many infected people will infect naive individuals, was estimated to be 2.2, with a confidence interval between 1.4 and 3.9. And only as we begin to do the serological surveillance will we get a better handle on this basic reproductive number to really understand if, as Michele is suggesting, will it have a bigger R naught and more people will be infected but will simply not display symptoms.
So this reproductive index is important because right now it's around 2, which is good. You know, a virus like measles is 15, which is really bad.
So every sick person infects on average 15 others.
Yeah.
The R0 is determined largely by the shedding period, right? The longer you shed and interact with other people. So that's why China right now is trying to find all the cases and limit them from interacting with other people. Because if you can cut the period, then you will cut the R0. If you can get it to 1 or less, then eventually you'll burn out the epidemic.
And that's why the quarantine has been put into effect, that the Chinese can actually suggest and force their people to stay in place.
Right. Although whether it's effective is another question or not.
Yeah,
you know.
People begin to head for the exits, as they say.
That's right. That's exactly right. So the good news is it's not a horribly serious. It's not an Ebola. It's not a horribly serious infection, and it should be we should be able to confine it, but it's going to go through a number of people because everyone in the world is not immune, or almost everyone in the world, unless you've been working with bats in the Chinese countryside.
I'd say the other bit of good news is that we do have an infrastructure in place to take what we've learned already about this new virus and then compare it to other similar viruses that have been described. So, for example, there is a Center for Structural Genomics of Infectious Diseases at Northwestern University that our colleague Carla Satchel is involved with, and they have money from the NIH to quickly mobilize and look at the structure of this new virus, compare it to other viruses that we know, coronaviruses, and enough detail that we know where drugs that are effective against these other viruses bind and inactivate the particular virus. So then you can deduce what other pharmaceutical tools we have that could be adapted or applied. So I'm just impressed. Like, this paper was published within just a couple of weeks of the first case.
Yeah.
And we have this Center for Structural Genomics, which already has been able to mobilize and get structural data and begin to make some deductions about where we should focus our drug discovery.
Yeah, it's great.
So,
and more importantly, Gilead has a clinical trial testing one of their existing nucleoside analogs that was developed for another virus, Ebola, but it may be effective in this particular epidemic outbreak.
Yeah.
Yeah. Another example, Vincent, the whole genome or the genome sequence of this virus was made publicly available in a matter of weeks also, right?
Exactly. Yes.
So there's great sharing and collaboration in the scientific community in response to this public health concern.
Yeah. Yeah. And many vaccine candidates are being developed now, so that won't happen immediately, but perhaps they will be available for the next one, because when this is controlled, there will be another one at some point. Another bad virus will jump into people, as long as we have these kind of wet markets that seem to be very popular.
Or as long as people are still on the planet. I mean, viruses spread person to person independent of wet markets. So, our planet is getting crowded, and so your likelihood of encountering someone who may not be displaying symptoms but may be shedding virus increases with the number of people on the planet. We're in our hospital. We're facing a run. We can't get masks for the infection control supplies.
Wow.
All the masks there's been a run on masks to Amazon. We've actually gone to Home Depot to look for examination masks, because Home Depot sells, masks for drywalling. And,
so, Michael, a surgical mask versus an N95, is there any difference?
Not for this virus, because it's likely droplet secretion.
So a surgical mask will take care of that, right? Surgical
mask. The N95 is if the person has some other bacterium that they're like to protect you against tuberculosis. But what the data still do not report is whether or not this is droplet secretion or whether it's truly airborne. Based on the R0, I don't think it's airborne, because we would have a higher R0 number, like measles is got a airborne component. It's not secretion. The virus is flying through the air.
I read in the New York Times today, Michael, that we do have a stockpile of masks and other respiratory protective gear.
That's in the emergency. That's in their emergency stockpile. Which
so
President,
what does it take?
It takes a release from the National Command Authority. That would be the president or the CDC asking for a declaration.
Okay. And so it's not at that level yet.
No, the fact is that the United States doesn't have person-to-person transmission at a rate that warrants the release of the national stockpile.
Right.
Yeah. So I have final thing I want to say. There's a wonderful live map of cases put up by Johns Hopkins. I'll put a link in the show notes. And there are currently 8,235 confirmed cases globally, 171 deaths. And the next highest country is Thailand with 14, Japan 11, Singapore 10.
And again, compared to 15 million flu cases this year.
Yeah, no comparison.
Get your vaccines.
No, get flu vaccines, yeah. Okay. If you want to hear more, listen to TWiM. We're going to be covering it every week for a few weeks. Okay. Elio, you got a paper for us?
I have a paper for you on a very strange disease. It's called Whipple's disease. And let me tell you what that's about. It's a rare disease. In fact, there have been no more than about 2,000 cases known since its announcement, which was in 1907. And guess who discovered it? It was Fred Hoyt Whipple, who was a famous medical scientist. In fact, he got the Nobel Prize for work on liver disease. Anyhow, Whipple's disease is interesting because normally it's considered to affect the gastrointestinal system and cause malabsorption. In other words, people with it do not absorb their food and they can starve. In fact, if untreated, they may even die. So the problem with Whipple's disease is that it does not only affect the gut, but also the brain, the heart, the joints, the skin, the lungs, etc. So it is a very odd disease, very rare, but because it causes so many symptoms, it is brought up in conversation, in medical conversation often. Maybe Michael can attest to that when people get together for clinical conferences to discuss rare cases, they do something called differential diagnosis. That is, they write on the board what it all could be. And sooner or later, Whipple disease shows up because it could be that.
And then they say, throw an antimicrobial at it, and if it goes away, you're cured.
Anyhow, Whipple's disease is caused by a bacterium. The bacterium was not understood because you couldn't cultivate it, and it's been identified by molecular techniques, and it can be grown in tissue culture, that is in cells in culture. The organism has been called Trophorema whipplei. Trophorema whipplei is a gram-positive member of the actinomyces, vaguely related to tubercle bacilli, not really, but in the same general group. The name Trophorema is interesting because trophe, as in trophic, is Greek for nourishment, and erema is Greek for barrier. So this is a barrier to nourishment. Now, this organism has been found not only in sick people, but in a lot of healthy people as well, and in the environment. So it's all over. You could even think of it as being part of the normal microbiome. It is that common. So it causes disease in very, very, very few people, and nobody knows quite why. So the big question comes up, is there a genetic predisposition to the disease? Is it possible that the people who come down with Whipple disease have some weakness in their immune system, something wrong that allows the organisms to thrive? And the answer is nobody knows. Except this paper, which is interesting because it has, let's see, how many, about 31 authors, if not more. I can't count that fast. the authors, what they found what is interesting here is that the authors, by the way, let me tell you the title is IRF4, haploinsufficiency in a family with Whipple disease. The first author is Guérin, the last author is Casanova, and they are mainly from France, but there's actually 4 continents. Asia, there's somebody from Qatar, Australia, somebody from an institute in Darlinghurst in New West South Wales, and from the United States, and Europe. So Europe, Asia, Australia, North America. It took 31 authors to do this. So what happened was they found that, oddly, in a family, there were 4 cases of Whipple disease. Okay? So this is the story. So what is it that they have in common? Well, they have in common the fact that they are deficient in a transcription factor involved in immunity called IRF4. So they had this variant of IRF4, which is known as R98W. They found that these carriers were young and heterozygous for R98W. So this was a loss of function. It modified the transcriptome of white cells following stimulation with the organism Triphoneema whipplei, the organism of Whipple's disease. So that's the story. So is it possible that this thing, which is known as a haploinsufficiency, that this lack of a proper gene for IRF4, transcription factor, is it possible that this is why they caught Whipple's disease? And it took an incredible amount of work to come up with the answer that they don't really know. Or, well, one can say that they do, that in fact this protein acts as a switch that turns on and off some genes involved in the body's response to infection. In patients with this mutation, the protein does not work as it should. So it may be caused by a specific gene mutation affecting subjects infected with T. whipplei. Okay, let's talk about the paper, and I think that Vincent will have more to say about it. But at the end, I'm going to tell you a little bit about another disease in cattle called Johne's disease, that's Johne, which is very similar to Whipple's disease in humans. I want to come back to that afterwards. But Vincent, do you want to pipe in on this?
Yeah, let me make a few comments. So this fellow Casanova, he's here at New York. He's at the Rockefeller. And this is what he does. He looks for people who develop severe disease, whether it be after herpes infection, influenza, even polio. He asked, why do some people get paralysis? Because it's only 1 in 100 people. And he tries to narrow it down to a mutation. And so this group of patients, very interesting. As you said, they have this single amino acid change in IRF4, which is a master regulatory protein of innate immunity. And this protein has a DNA binding domain, which binds the DNA of the promoter. And this single amino acid change blocks DNA binding. It's quite interesting. And they show, as you said, that in cells with just a heterozygous, 1 copy of the DNA has this mutation, it's enough to change the transcriptional profile. In other words, the genes that are turned on by this protein, which is weird because there's still a normal copy of the gene in these cells. And that's what haploinsufficiency means if you have 1 mutated copy of the gene, there's still a problem. And we don't know why. One of the things
How common is this, by the way?
Haploinsufficiency?
Yeah.
I don't know the answer, but it's a well-known genetic phenomenon, right?
But I think it's rare, isn't it? I mean, mostly you need to have a diploid insufficiency.
Well, usually you have diploid, or if 1 copy may have a dominant negative effect.
That's right.
Right?
That's right.
And they actually did experiments to address that, and this altered protein does not have a dominant negative effect. Right. Yeah. So somehow it's messing up. I mean, a simple explanation is that there is not enough protein. You know, you need both alleles cranking out mRNA. And
it's kind of strange. I mean, it's just a factor of 2.
It's only 2, exactly. So it's kind of a puzzle. And
it is a puzzle, I think.
But I think that the data are beautiful. And I mean, they are really convinced that this is behind this disease. But one thought I had, okay, so IRF4 is really important. Why don't we see other infectious diseases associated with people with this mutation, right? You would think maybe.
That's right. The curious thing is these 4 cases don't have sensitivity to other infections.
Yes.
That's totally normal with regard to other infections. It's only Whipple.
It's very odd.
Yeah, and we don't know that much about the pathogenesis of this microbe to begin to make hypotheses about why that is, do we?
Well, what is known is that in the intestinal form of the disease, the macrophages, which are underneath the epithelium, become very strange. They are big and swollen, and so
foamy. Yeah,
it may be something that the organisms do to these macrophages. It's not a very good answer. By the way, the effect of that is that the villi in the intestine become swollen and misshapen, and that accounts for the malabsorption.
Right.
So it sounds like this particular IRF4 is really specific for this disease, unless they find it elsewhere. It's specific. And so in certain cells that maybe, as Michele said, that are important for the pathogenesis, maybe this particular function is important. So clearly more work is needed, but this is Elio, my question is, now that we know this, can we use this knowledge to treat these patients?
To vaccinate them even.
Well, treating patients of Whipple is not a big problem because it's sensitive to antibiotics.
Okay.
So you have to do treatment for a long time, but it's very effective. So, that's...
The other thing I wanted to point out is that this mutation is not 100% penetrant, right? Not everyone with heterozygous R98W will develop Whipple.
Right.
And
so that makes it even more difficult.
Yeah, which is not so unusual in the infectious disease field. In fact, colleagues Arturo Casadeval and Lisanne Perovsky have been for the last 20 years putting forth this idea that it's almost a misnomer to say that this is a beneficial microbe, whereas that is a pathogen, because there are so many factors that go into dictating what the outcome of any one interaction is. And they've described this as a damage response framework as kind of a broader way to think about pathogenesis. So, for example, the size of the inoculum will determine who gets sick and who doesn't. Yeah, sure. What the temperature is, the age of the person affected, their own personal history with related bugs or other infections. So yeah, it's not just a one-to-one relationship between if you're exposed to this bug, this is what the symptoms are. And of course, we discussed that with the new coronavirus that was also illustrated there.
Yeah.
It's amazing that they were able to take advantage of this one family and find this really specific mechanism that accounted for the disease. I'm impressed.
Yeah, that's often why these in the other papers from this lab, from Casanova, they often have many, many authors because a lot of them are involved in recruiting the patients. To the study. Yeah.
Right.
It's everything about it is strange. So let me bring up the fact that there's a very related disease in cattle, Johnes disease. Johnes disease is well known among veterinarians because it causes the same symptoms as the intestinal form of Whipple's, namely malabsorption in cattle, and they starve. So Johnes, which is an old disease, is sort of the equivalent of Whipple's in humans. It is caused by a mycobacterium, subspecies paratuberculosis. In other words, it's also in the actinomycetes. So vaguely, the organism that causes Whipple's and the organism that causes Johnes have some vague, they're in the same general group of the actinomycetes. The Whipple organism is not acid fast, but it's not that far from being in the mycobacteria. So maybe there's a connection there, maybe there isn't. Now, the reason I have a soft spot for Jonas, let me tell you my little story. I spent most of my life, my career at Tufts University. And one good day, the president of the university decided to found a veterinary school. There wasn't a veterinary school in all of New England. That was fine with me. Why not have a vet school? But they decided that the basic science departments in the medical school, and I was in one of them in microbiology, would be in charge of teaching the vet students. Well, that caused a little problem because I rapidly found out that the diseases, the infectious diseases of animals are not exactly the same as those of humans, even though we catch certain diseases from animals. So we were given the money to hire new people. We did, etc., etc. But I read up on it, and I ran into Jonas disease long before I heard of Whipple's, and I was really fascinated. So I read up on it. It's something that caught my eye, and it was worth my attention. So one good day, a group of veterinarians came to certify our ability to teach vet students. We needed to be accredited. So a group of vets, senior vets, came and talked to us, and they asked me what did I know about animal infectious diseases, and I proudly trotted out Jonas, which surprised them because it's not the first thing you think of. So here I was, ready to deal with Jonas disease, and they kept changing the subject, and I kept bringing it back to Jonas.
Great.
So it reminds me of the kid who's working on his school assignment, and his school assignment is about animals, so he reads up all about the snake. Goes to school, what does the teacher say? The teacher says, let's talk about the elephant. So the kid says, the elephant has a snout that looks like a snake. Now the snake, I did exactly the same thing with Jonas. Now Jonas disease.
Good.
And they still accredited the school.
They accredited us. Guess
Yes.
what? Accredited us. So that's my story.
So we don't know in Jonas if the cows have an IRF4 mutation, right?
We don't know anything about Jonas.
Mm-hmm.
No,
But cows are likely more inbred than people are because cows have been selected for the quality of their milk, the marbling of their meat, and all sorts of other traits that we use to select the perfect animal.
Yep.
So I imagine that this paper that we're discussing may have an effect on the veterinary world. That they may start looking at the cows a little bit closer.
Well, you know, the genomes of livestock are being sequenced as much as people because it's such a vital economic resource, and understanding what makes the perfect cow or makes the perfect pig is very much in the forefront of animal husbandry.
Well, thank you, Elio.
Sure.
We have a couple of interesting emails. Let's see, get to a couple of them. The first one is from Allison. Dear TWiM, I've never written into things before, but I wanted to write and thank you for the countless hours of free education and weather updates. I'm presently in a community college for an associate's diploma in medical laboratory technician, so I'm literally a greenhorn. I began listening to your podcast in the summer to get me in the right headspace back into medical science, particularly microbiology. I'm glad I did. I went back to a lot of earlier shows, devoured them eagerly. You explain things very well and go over so many different concepts, some of which I had begun to learn myself during my education, from the basics of antibiotic resistance, cellular respiration, and basic biology to some fascinating topics like the development of bacteriophages and how they may be possibly tailored to an individual's need, and the Asgard archaea and the work it took to discover and research these new prokaryotes. Not prokaryotes.
We're going to get a letter from Mark Martin.
I'd even
or
use
Norm Pace.
Well, explain yourselves.
Well, archaea are archaea. They are not prokaryotes or bacteria, right?
Correct. They're in a different kingdom, part of the three kingdoms. The eukaryote is a old word which means it has a true nucleus. A prokaryote means without a nucleus. And as we know, the archaea also do not have a nucleus, but they are in a separate kingdom from the bacteria. So they're now referred to as bacteria.
Archaea.
Or microbes, if you want to.
Or microbes.
General.
Eukaryotes, bacteria, archaea, the three kingdoms. Sorry, Allison. Okay, I had even used the case you presented in your second episode about the researcher who contracted septicemic plague from an attenuated strain of Y. pestis as case study to my peers as well as high school students in our region.
Good for you. That's great.
As I continue to learn this new world, I can't help get a little excited when I find myself able to understand the topics that are brought up. Your explanations are clear, your metaphors are always fun and really help those new with a casual interest in microbiology. Textbooks are fine tools, but learning through current topics or discoveries really colors the world beyond what a class can. Nice.
Well, that's nice.
Mm-hmm.
I'm hoping to continue to pursue an education in microbiology, particularly epidemiology, and looking into this world is still daunting. Is there any advice you can give to bright-eyed new future microbiologists and the best way to approach a future career in it? Thank you all again. Hope you have a wonderful 2020. P.S. Every time I see mushrooms or drink wine, I think of Elio.
All right.
Hey!
Mushrooms.
Thank you.
Any advice?
I'll suggest, Allison, that you look out for people that are working in fields that you're interested in and just ask if you can sit down and talk with them, learn about their path and any advice they might have or opportunities that they're aware of. So don't hesitate to reach out to other people and have conversations.
One of the only bright spots of this outbreak of coronavirus is to illustrate the importance of epidemiologists and their extremely important role in keeping our planet safe. So I think as you begin to follow the coronavirus outbreak, you can really see a place for yourself as you continue to delve into this brave new world of the microbial kingdoms and worlds that you've been approaching.
Yeah, but talk to some of the people that are teaching your classes in the your medical lab technician program, and I'm sure they'll be able to introduce you to others that can set you on your way.
All right, we have one from Trudy. Dear TWiMers, over the holidays, I had a lengthy discussion with a friend of mine about whether wooden cutting boards and spoons should go into the dishwasher. I recall a similar discussion on a previous TWiM, with the conclusion being that while the dishwasher does sanitize these items, it dries them out, creates cracks, and consequently more spaces for microbes to grow. Or maybe I'm not remembering correctly. Could you please advise? Thank you and best regards. Michael, you know all about cutting boards, right?
I do unfortunately know about cutting boards. Good wood cutting boards require good oiling with food quality vegetable oil. And the oil causes the wood to effectively seal those cracks and prevent the microbes from the ones that cause infections of people. It prevents them from getting a foothold, if you will, because the oil will hydrate the wood, and that actually serves to preserve it, and a good oiling of your cutting board will help things out. But as a rule of thumb, if you're preparing fresh poultry, it's best not to use your good wood cutting board. It's better to use something that you can put into a dishwasher so that you don't run the risk of allowing those nasty coliforms or other enterics a foothold in your good wood cutting board.
But I use wooden spoons, wooden salad bowls, and of course salads typically are just made with washed vegetables. So I certainly don't put them in the dishwasher. I just give them a quick rinse, wipe them dry, and I don't even oil them. Michael, do you oil your wood spoons at home?
Normally there's enough inherent I don't want to use the term grease, but there are enough oils in the foods that we cook with when you're sauteing using the wooden spoon that generally serves to seal the cracks.
Yeah, and same with your salad bowl. Your vinaigrette's got enough oil.
Yeah, the vinaigrettes have both vinegar as well as oil. So the vinegar will inactivate a large number of the coliforms and enterics, and the oil will serve to seal any of the incident cracks that develop.
But be more careful with chicken.
Yes, chicken. You got to make sure chicken and hamburger are the spooky ones.
So basically, if you happen to put your wooden stuff in the dishwasher, you should oil them afterwards.
Well, they're sterile at that point in time, or near as close to being sterile after the sanitation cycle really cooks them. But they will develop cracks as they dry out. Okay. It strips the oils off of them because of the heat and the soaps. Breaking news, WHO has declared a global health emergency on the corona. That means more resources will be available to everyone to be able to do something. Well, that's actually good news because it will link all of the epidemiological surveillance. People will be following it more closely at local health departments and national health departments, and the globe will get a better overview of how this virus is spreading, and it will reinforce the need to get a vaccine against influenza, which this year is pretty nasty. It seems to be Group B, and it does indeed cause illness in people. People are reporting that they have been vaccinated, but they're nearly as sick as they could have been if they hadn't been vaccinated. So I also hope that some good news from this response to coronavirus will strengthen our public policy and our infrastructure for dealing with these sudden new emerging pathogens, because as Vincent pointed out, there will be more. You bet. And one more email from Ryan. Dear TWiM, there's a story out there of a person from Minnesota who has been given phage therapy. This is from the Chicago Tribune. Interesting story where this man was going to have a leg amputated. I think he had an infection in his knee. They decided to try phage therapy. There's a company in Maryland called Adaptive Phage Therapeutics. They had done, 13 previous phage treatments, and they treated this man, and he still has his leg. That's good news. Yeah. I guess they have a bank of some 1,000 different phages, so they're trying to probably match them up with the particular infectious agent in each patient. And they say some clinical trials are underway at 10 different sites. Yes. So it's exciting. Can I ask you a question? Yeah. I'm looking at this breaking news in the New York Times. The WHO declared a global health emergency. One of the things they say, it serves notice to all United Nations member states that the world's top health advisory body thinks the situation is grave. Is that justified? Is this a grave situation? No, I don't think it's any more grave than influenza, as I speak. Exactly. Or the vaping issues that we're seeing in the United States. Right. It certainly unsettled the stock markets. Oh, yes. Just curious. Yes. It did the other day, yesterday. Yeah. We'll see. It has economic repercussions. Apple is saying they won't be able to make enough iPhones, for example, because they're all made in China. So I don't think it's grave, but that's the language WHO uses when they declare an emergency. I don't know. Maybe it's in part to convince nations that they should pay attention. I don't know the politics behind it. I also don't want to downplay the stress it's putting on the people in China, that they're not being allowed to travel. They're shutting down major transportation avenues. So it's disruptive for sure. All right, stay tuned. We will keep you posted. Go over to TWiV for a weekly update. TWiM is another one of our podcasts. You can find it on any good podcast player. We ask you to subscribe so you get every episode, and we know how many people are listening. That helps us in our funding efforts. And speaking of funding, if you like what we do, consider supporting us financially. You can go to microbe.tv/contribute, and you could do things like give us $1 a month or a small amount of money, whatever you'd like, to help pay our expenses. And if you want to ask questions, give comments, TWiM at microbe.tv. Michele Swanson's over at the University of Michigan. Thank you, Michele. Thank you. I learned a lot. How's golf going?
Don't tease me.
Just kidding. Couple months, right? Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure. Thank you for having me.
Always. Wouldn't not have you ever.
Okay.
Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm proud to have all three of you. It's my pleasure and honor to be able to do this with you. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM. Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.